Alright guys, so I hope you had a good time with your project, doing something a little bit different. We are going to go ahead and get back into our regular note material, and we're going to finish up this unit with another type of cell division called meiosis or meiosis. And so I want to go ahead and start by saying I do say it incorrectly. It's supposed to be meiosis, but normally when I teach about it I always say meiosis instead because it sounds a little more different from mitosis. So you can say it however you want to. The main thing is this is going to be a different type of cell division than what we saw with the cell cycle. And so in the cell cycle, what we were making were somatic cells, and soma is a prefix that means body. And so somatic cells are just your regular old body cells that make up all of the different organs and tissues in your whole body. Pretty much it's going to be your blood cells, your skin cells, your nerve cells, your muscle cells, all of those. And all of these cells are cells that have a complete set of chromosomes, and the word that we use to describe that is diploid. And we need to get really comfortable with a couple of terms in this set of notes, and that's going to be diploid and haploid. And so if a cell is diploid, what that means, it has two sets of chromosomes, and we represent that by 2N. N represents a set. Now the reason we have two sets is, remember, you are half your mom and half your dad. So your mom gave you 23 chromosomes and your dad gave you 23 chromosomes, and that's what gives you a total of 46 as a human with a normal chromosome number. And so all of the cells in your body, pretty much with one exception, they're going to be diploid body cells or diploid somatic cells. And these were the cells that, if they are able to, they're going to go through the cell cycle. They're going to divide by mitosis and cytokinesis, and these are the ones that we're making new that are the same. Now, if we tried to reproduce with those and two somatic cells were joined, what would happen is you would end up with 92 chromosomes, and that's clearly not our normal chromosome number of 46. And so what has to happen is we have to have a different type of cell for sexual reproduction. And that is what meiosis is all about. Now we call meiosis a reductive division, and the reason why is because we're going to be reducing the number of chromosomes. And so instead of producing somatic body cells, meiosis is going to produce gametes, and that's just a fancy word for sex cells. Now instead of being diploid, remember that two diploids are not going to give us a human. Instead of being diploid, meiosis is going to create these sex cells that are haploid. And so diploid meant two sets of chromosomes, so haploid is going to be one set or half. Okay? And so this way, when half and half come together, we get a whole. So your egg cells for the females and your sperm cells for the males are all going to be haploid. They have half the number of chromosomes. So this way, when fertilization occurs, mom is going to contribute 23, dad is going to contribute 23, and that produces the offspring, which has two sets or a diploid number of 46. And so before we get into the processes of meiosis, just a couple of terms. Now, chromosomes we have seen before. I just want to remind you that this is the condensed form of DNA. We see it during division. And I just want you to remember that when you see the X, it's a duplicated copy. Okay, so remember, we start with singles. In the S phase of interphase, we double our DNA. And so by the time we see the chromosomes, they are X's and they are double copies. They each have two sister chromatids held at the centromere. Now, another important term is something called homologous chromosomes. Now, the prefix homo means the same. Okay, I was trying to do quotation marks here. There we go. So the prefix homo means the same. And so homologous chromosomes are chromosomes that are the same from each parent. Now, when I say the same or that they are alike, they are the same in that they are the same number chromosome. So it's like chromosome number 1 from mom and chromosome number 1 from dad. Chromosome 5, chromosome 5. Chromosome 20, chromosome 20. Okay? So they're the same number, and when chromosomes are the same number, that means they are the same size and they have the same genetic info on them. Now, I do not want you to think that they are identical. They are not identical. And we haven't really gone into genetics too much yet, but they have the same genes, but they may not have the same alleles. So for instance, let's say, and I'm just making this up, but let's say that chromosome 4 had a gene for eye color. Now, both chromosome 4 from mom and chromosome 4 from dad are going to have that same gene for eye color. But where mom said blue, dad could say brown. Okay, so that's what we mean by different alleles. So they're not genetically identical. They're just the same in their size and the genes that you find on them. Okay, so these are going to be your chromosome pairs, your 1s, your 2s, your 15s, all the way to 23. And remember, you get 1 from mom and 1 from dad. Now, in meiosis, there's going to be some distinct differences from mitosis. And one of the big differences is that in meiosis, there's 2 divisions. And we literally call them meiosis 1 and meiosis 2. Now, because we divide twice, we're going to get 4 new cells. And because of some processes that happen within meiosis 1 specifically, our cells are going to be different. They're going to be unique, and they are going to end up being haploid. So this is a very different outcome from what we saw in mitosis in the cell cycle. Okay. Now, where this is happening in the body, for females, this is happening in the ovaries, and for males, this is happening in the testes. Now, the cells that make up the ovaries and the testes are going to be somatic body cells, just like every other organ. But in particular cells that we will find in the ovaries and testes, they are going to go through these 2 divisions to create the gametes. Now, I know this may not make sense just yet because we haven't talked about the processes, but this little section right here is really important to understand, okay? Meiosis 1 is going to be separating the homologous chromosomes, or those pairs. Meiosis 2 is going to be separating sister chromatids. So the events that happen within meiosis 1 and meiosis 2 are not going to be exactly the same. And something I want you to maybe make note of, and we're going to talk about this later, is that meiosis 2 is going to look just like mitosis. It's going to be very similar to what we've already studied. So most of the differences are going to come in meiosis 1. And before we get into the divisions, we're going to have an interphase just like we saw before, okay? So there's only going to be one interphase. So prior to meiosis 1, there will be an interphase with G1, growth, S, DNA replication, G2, more growth and prep for division. So just like we saw in the cell cycle, the cells that are in the ovaries and the testes that are going to go through meiosis, they are going to go through an interphase. And so that DNA is going to be copied before we divide. But this time, we're going to divide them twice. Okay, so 1 interphase, 2 divisions. And so we're going to start with meiosis 1. And meiosis 1 is where we're going to see some key differences from mitosis. And we're going to talk a lot more about meiosis 1 than we are meiosis 2, because meiosis 2 is going to be very familiar. So in meiosis 1, prophase 1, and I want you to note how we put a number after them. Okay, so this is how you can tell whether it was mitosis or whether it's meiosis, because if it's a meiotic stage, you're going to see a number following PMAT. If it's mitosis, it's just prophase, metaphase, anaphase, telophase. Okay, so in prophase 1 of meiosis 1, we're going to have your typical prophase event. So this right here is all the same stuff that happened back when we saw mitosis, prophase. Now, the key difference is going to come in that there's going to be some extra events that happen in meiosis 1 that do not happen anywhere else. And something that's unique is that chromosomes are going to line up, or they're going to pair up with their homologue, okay? This did not happen in mitosis. And so what happens is your pairs pair up. And when they're together, they're called a tetrad. Okay, tetra means 4, and when they're paired, there are 4 chromatids, so that's where that's coming from. So your number 1s find each other, your number 5s find each other, your 12s find each other. And this is going to happen for all of your homologous pairs, all of your like chromosomes. They're going to find each other and pair up in a process called synapsis. Okay. Now, while they are paired and while they are synapsing, they may exchange pieces of their chromatids, and this is called crossing over. Not every chromosome pair will do this, but if just one does it, we're going to have unique gametes, okay? And so what can happen with crossing over is when they pair up, when they synapse, they kind of join and kind of overlap each other at a location called the chiasmata or the chiasma. And in the process of crossing over, they can literally just exchange pieces of their chromatids. Now, remember that your homologous chromosomes are the same size, they have the same genes on them, but they are not identical. Okay, so let's say this was dad chromosome, and let's say that this was mom chromosome. What can happen during prophase 1 is they kind of pair up in the process of synapsis. They may overlap their little chromatids, and they can literally flip-flop the same piece, okay? And so the significance of crossing over is that it leads to variation. If just one pair of our 23 pairs cross over, every single one of our egg and sperm are going to be unique. And so this is why you are not identical to your brothers and sisters. The only identicals are going to be identical twins. But every time your parents have a child, they are going to have unique combinations of egg and sperm because of crossing over, and another process that we'll see here in just a minute, okay? So what I want you to notice is that if crossing over occurs, now no two chromatids are alike. So there's chromatid number 1, chromatid number 2, chromatid number 3, chromatid number 4. Now, when they go to divide these up in meiosis, each one of these chromatids is going to end up in a different sex cell. And so now every sex cell is going to get a unique combination. So that's just if one chromosome pair crosses over. So imagine if you have multiple chromosome pairs crossing over, that's just going to increase the diversity in your final gametes. Now, up next is metaphase 1. And this is going to be another phase where we see a key difference from mitosis. Now, they are still going to line up in the middle. Okay, meta still equals middle, but this time they're going to line up as pairs or tetrads. They're going to line up with their buddy. Okay, in mitosis, they lined up like this. In metaphase 1 of meiosis 1, they're going to line up as doubles. Okay, so they're lining up with their buddy, with their homologue. And we also have another source of variation here in a process known as independent assortment, okay? So you can see here, let's say that green represents mom and blue represents dad. So this pair lined up like this. Now, they could have just have easily flip-flopped. So when they line up as pairs, all 23, you're going to get a unique combination of mom and dad chromosomes, because it's not like a certain one has to go on a certain side each time. So this is what we call independent assortment. It's a random lineup of tetrads, and this leads to a unique combination of maternal and paternal chromosomes. All right, so now in anaphase, because they lined up as pairs, we are still pulling them apart, but this time we are separating homologous chromosomes. And so a whole chromosome is going that direction, a whole chromosome is going that direction. And then in telophase, we have the same events that we had before. The nuclei are going to reform, spindle's going to break down, and then that's going to be followed by cytokinesis that divides the cytoplasm. So because of those events that occurred in prophase 1 and metaphase 1, we're going to end up with a very different result from what we saw back in mitosis, okay? Now, we are going to have two cells, but they're no longer going to be identical. They're going to be different because of crossing over and independent assortment, and they are also going to be haploid. We are already to a haploid chromosome number. Now, here's the problem. We're not done yet, because even though the cells are haploid, the chromosomes still look like an X. And hopefully you remember that when we see the X, we know that it is a double copy, okay? So the next stage is going to be all about going in and dividing those duplicated chromosomes. And so this is why meiosis 2 looks just like mitosis. Okay, the only difference is we're starting with haploid cells, but the processes of PMAT are going to look just like they did to mitosis. Okay, and what we're doing is we're separating the sister chromatids just like we did in mitosis. And so you can see here we're not going to spend nearly as much time going through the phases. We have normal prophase stuff, nucleus is breaking down, chromosomes form. At this point though, remember we have half the number of chromosomes. So they're all duplicated though. In metaphase they're going to line up, but they're going to line up individually just like they did in mitosis, single file. In anaphase we're going to pull apart the sister chromatids just like we did in mitosis. And in telophase 2 we're going to get our new nuclei back, and then because of cytokinesis they will fully divide into 4 new cells. And so by the end of meiosis 2 I have 4 cells because they were 2 divisions. They are different because of independent assortment and crossing over, and they are haploid because of when they line up in pairs and we separated the homologous chromosomes. So the end result looks very different than what we saw back in mitosis. Now, sometimes mistakes can happen, as with any process, and one mistake that can occur in meiosis is a mistake known as non-disjunction. And so sometimes when the chromosomes are being separated, this could happen in anaphase 1 or anaphase 2. So when those chromosomes are being pulled apart, sometimes they are not separated properly. And so what happens is some gametes from that meiotic division end up with missing chromosomes, and some gametes end up with extra chromosomes. And this leads to something we call aneuploidy, okay? So if these gametes that either have missing or extra chromosomes are used to produce offspring, it's going to lead to an abnormal number of chromosomes. Now, we have a few different common disorders that result from this. Probably the most common is Down syndrome. This is where the offspring receives an extra 21st chromosome. Turner's is where it's a female, but there's only one X, okay, instead of 2 X's. Klinefelter's is a male that has an extra X. There's some others like Edwards and Pateau syndrome. Now, if you have an abnormal number of sex chromosomes, it normally doesn't affect survival. So like Turner's and Klinefelter's, there's going to be some effects, but it doesn't necessarily affect their ability to live. Now, you don't hear about a lot of these abnormal chromosome numbers on other chromosomes. Like I've never heard of an extra or missing chromosome 1 or 2 or 3. And that's because a lot of times if you have abnormal chromosome numbers on these 1 through 22, a lot of times the effects can affect development and the baby never survives to recognize that disorder. Now, something else to note is that this non-disjunction, it does increase with the mother's age. So the older the mom is, the greater the chance that they could have a baby that does have a chromosomal abnormality. And one of the reasons for that is because meiosis is a little bit different in males and females. So if you're a dude, this starts when you're at puberty, and pretty much you produce sperm until you die. Okay, there could be some things that could affect sperm production, but for the most part, at puberty, guys start going through meiosis and they become sperm factories, and every time meiosis happens, potentially all 4 of those sperm can be used to create a baby. Okay, now with females, this actually starts before you're even born. Meiosis is a kind of started and then arrested in the womb. So you pretty much go through meiosis 1 while you're developing in the mother's womb, and then you will complete the meiotic process and kind of continue once you hit puberty. And with females, by the time you get to puberty, you've already lost a lot of those potential egg cells. The division will continue. 1 egg will kind of be chosen a month typically. But what happens is we don't get all 4 cells produced that could be used to make babies. From the very beginning, the mom is the nurturer. And so whenever they're dividing in cytokinesis, 1 cell gets way more of the cytoplasm and the organelles. And this is the one that we call the egg, okay? The other 3 cells, they get the chromosomes, but they don't have all the other stuff. We call these polar bodies, and they're not really usable in reproduction. So where guys start at puberty and get 4 sperm each time, females start in the womb and only get 1 reproductive egg each time meiosis completes. Now, some other things, this is just going to be a quick review. So meiosis is all about sexual. We are producing the gametes needed for sexual reproduction. We know that these gametes are unique, and we take gametes from 2 different parents, and then through the process of random fertilization, we create a zygote, and then this zygote is going to be different from the parents, okay? And so genetic diversity is a really good thing. It increases chances of survival for natural selection. And then in asexual, which is what we saw like with mitosis in the cell cycle, remember there's just 1 parent, we get identical offspring, and so we don't get that variation. So really sexual reproduction is superior because we get that diversity. And so we're going to end with just a quick side-by-side comparison with mitosis and meiosis. Now they both start the same. They both start with a diploid somatic body cell. They both go through an interphase and DNA is replicated once. But at this point in mitosis, there's 1 division, PMAT. In meiosis, there's 2. The end result is very different. Mitosis gives us 2 identical diploid cells. Meiosis gives us 4 different haploid cells. And that's because of those events like crossing over and independent assortment. Those did not happen in mitosis. And mitosis and the cell cycle are used to create new somatic cells, whereas meiosis is used to create sex cells or gametes. And that is going to be all for your meiosis notes. So a few new things to learn. We're going to spend a couple of days practicing, and then we're going to be getting ready to review and take a test. If you guys have any questions, don't forget to ask your teachers or email me, and hopefully I will get to see you soon. Hope you all have a good day.